Good morning, everyone. I am delighted to welcome you all today. Today, we will be exploring the fascinating world of high-performance thin-layer chromatography, or HPTLC. I'm Dr. S.K. Singh, and I'll be guiding you through this presentation. We'll cover the fundamentals of HPTLC, its applications, and why it's such a powerful tool in analytical chemistry. I look forward to sharing insights and engaging discussions with you all. Let's get started. Welcome to today's discussion on HPTLC, or high-performance thin-layer chromatography. As you can see, HPTLC is actually an advanced version of the traditional thin-layer chromatography, or TLC. It's designed to provide more precise and reliable results. This method is used both for qualitative analysis, which means identifying what compounds are present, and quantitative analysis, which involves measuring how much of each compound is there. HPTLC has a wide range of applications. You'll find it commonly used in the pharmaceutical industry to analyze drug components, in herbal medicine to verify the authenticity and quality of herbal drugs, and also in food and environmental testing to monitor contaminants or nutrients. What makes HPTLC stand out compared to TLC is its higher resolution, meaning it can distinguish between compounds that are very similar. It also offers greater sensitivity, so it can detect smaller amounts of substances, and it provides more reproducible results, ensuring consistency across different runs. Overall, HPTLC is a powerful tool that enhances our ability to analyze complex mixtures efficiently and accurately, supporting better research, quality control, and safety assessments across multiple fields. Let's dive into the basic principle of HPTLC. Essentially, this technique relies on the separation of components based on their differential adsorption between two phases, the stationary phase and the mobile phase. To put it simply, different compounds will interact with these phases in unique ways. Because of these differences, the components don't all move at the same rate when the mobile phase travels through the stationary phase. Instead, each component moves at its own pace, which leads to their separation on the plate. This movement is quantified using what's called the RF value, or retention factor. The RF value helps us identify and compare compounds by representing how far a component travels relative to the solvent front. So, in summary, the core idea here is that separation in HPTLC hinges on how differently compounds adsorb onto the stationary phase versus how they are carried along by the mobile phase. Understanding this concept is fundamental to interpreting the results correctly. Let's take a closer look at the components of the HPTLC system. First, we have the stationary phase, which consists of pre-coated plates made of materials like silica gel or alumina. These plates serve as the foundation where the separation occurs. Moving on, the mobile phase is essentially a solvent system that moves through the stationary phase, carrying the sample components along. Next, we have the sample applicator, which is often automatic, ensuring precise and consistent application of samples onto the plates. Then, there's the development chamber, where the plates are placed to develop the separation under controlled conditions. Once the separation is complete, detection becomes crucial. The system uses a detection method such as UV light or derivatization to visualize the separated components. Following that, we have the scanner or densitometer, which measures the intensity of the signals and helps quantify the components. All these parts work together seamlessly, making HPTLC a powerful and efficient method for analyzing complex mixtures. This integrated setup highlights the precision and reliability of the system, which is essential for accurate analysis in our overall workflow. Let's take a closer look at plate technology, which is at the core of this method. HPTLC plates, for example, feature smaller particle sizes, roughly around 5 micrometers, which helps in achieving better separation. They also have a uniform coating, ensuring consistent quality across the plate. Now, why is this important? Well, these improvements translate into several advantages. We get better resolution, meaning we can distinguish between compounds more clearly. The bands we observe are sharper, making analysis more precise. Plus, the lower detection limits allow us to identify even trace amounts of substances. Moving on to the principle of separation, it primarily relies on mechanisms like adsorption, where analytes stick to the stationary phase, partition, where compounds distribute between phases, and in some cases, ion exchange, which involves the exchange of ions between the analyte and the stationary phase. Several factors influence how well this separation works. The polarity of the analyte plays a big role. More polar compounds interact differently with the stationary phase. Solvent strength affects how substances move through the system. The thickness of the layer also matters. A thicker layer can change separation efficiency. Lastly, chamber saturation is crucial. It ensures the environment is consistent, which helps in achieving reproducible results. By understanding these core principles and factors, we can optimize the technique for various analytical needs. Now, let's talk about chromatographic development. When we develop a plate, it's typically placed in a saturated chamber to ensure consistent humidity and conditions. The mobile phase then rises through the plate by capillary action, carrying the sample with it. This process can be repeated multiple times if needed, which helps improve the separation of compounds. Controlled conditions, of course, are essential because they enhance reproducibility, making sure our results are consistent across different runs. Moving on to the sample application principle, it's important to note that samples are applied as bands, not spots. Using automatic applicators is a key advantage here because they provide high precision and reproducibility. Plus, applying the sample as bands minimizes diffusion, which is crucial because it leads to better separation of the components. Overall, these techniques and tools work together to give us more accurate and reliable chromatographic results. Now, let's talk about quantification using densitometry. The scanner measures the intensity of the signal and relates it to the concentration of the sample. This process allows us to generate a calibration curve, which is essential for accurate quantitative analysis. With this, we can analyze multiple samples simultaneously, saving time and increasing efficiency. Moving on to detection principles, there are several methods we can use. Ultraviolet detection at 254 nm or 366 nm is common, as well as fluorescence detection, which is highly sensitive. Chemical derivatization is another technique that can enhance detection or help with identification. Visualization plays a key role here too. It enables fingerprinting, which is useful for identifying specific compounds, and identity testing, confirming what a sample actually is. Together, these methods and visualization techniques form a comprehensive approach for detailed analysis and accurate identification in our overall workflow. Now, let's focus on method validation, specifically how it aligns with ICH Q2 compliance. When we're validating an analytical method, there are several key parameters we need to consider to ensure our method is reliable and reproducible. First up is specificity. This is about making sure our method can accurately identify and quantify the analyte of interest, even in the presence of other components. It's crucial because we want confidence that our results are truly representative of what we're measuring. Next is linearity. This parameter assesses whether our method produces results that are directly proportional to the concentration of the analyte across a specified range. It's fundamental for quantification accuracy. Then we have accuracy. This measures how close our results are to the true value. Ensuring accuracy means our method is providing correct data, which is essential for regulatory acceptance. Following that is precision, which refers to the consistency of our results when the method is repeated under the same conditions. It includes repeatability and reproducibility, and it's vital for demonstrating reliability. We also need to consider the limit of detection, or LOD, and limit of quantification, or LOQ. These parameters tell us the smallest amount of analyte we can detect and quantify with confidence. They are especially important when dealing with trace levels. Finally, robustness. This assesses how resilient our method is to slight variations in experimental conditions. A robust method ensures consistent performance even if some parameters change slightly. All of these parameters are mandatory for regulatory acceptance because they collectively demonstrate that our method is valid, reliable, and fit for purpose. So, when we're validating our analytical methods, keeping these key points in mind helps us meet regulatory standards and ensures the quality of our data. When we look at the advantages of this method from a regulatory perspective, one of the key points is high throughput. This means we can analyze many samples at once, which is a huge time saver and boosts efficiency in our workflows. Another important benefit is low solvent consumption. This makes the method more environmentally friendly, aligning with green chemistry principles, and also helps reduce costs. Speaking of costs, it's a very cost-effective approach, which is always a plus in any analytical setting. Plus, this method is particularly suitable for herbal fingerprinting, allowing us to obtain detailed profiles of complex herbal mixtures with confidence. And finally, it fully complies with QBD principles, or analytical quality by design, as outlined in ICH Q8 to Q10 guidelines. This means the method is robust, well understood, and designed for consistent quality, which is crucial for regulatory approval and ensuring reliable results. Overall, these advantages make this approach not only scientifically sound, but also practical and aligned with current regulatory standards. Thank you for your attention as we wrap up. HPTLC, or high-performance thin-layer chromatography, is truly a robust technique that we can rely on for consistent and reproducible results. It's also widely accepted by regulatory agencies, which is crucial for its application in various industries. As we've seen, its role is expanding in key areas like quality control, where it helps ensure product consistency, stability studies to monitor how formulations hold up over time, and herbal standardization, which is vital for verifying the authenticity and potency of herbal products. Looking ahead, I see exciting possibilities with the integration of HPTLC with other advanced techniques, such as hyphenated methods like HPTLCMS. This combination will enhance our analytical capabilities even further, providing more detailed and comprehensive data. Overall, HPTLC's versatility and future potential make it an invaluable tool in our analytical toolbox. For further support, please don't hesitate to contact us. We're here to help you with any questions or additional information you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're here to support your ongoing success, so feel free to reach out whenever you need. Thank you again, and I look forward to connecting with you soon.